Welcome everyone to the second NeuroMac webinar. And our speaker today is Dr. Alexi Nott. So just shortly introduce him, he completed his PhD at the UCL, University College London, with Dr. Riccio, where he investigated the regulation of HDAC2 by nitric oxide signaling during the brain development. He then moved to the Psi Lab at MIT for his postdoc, where he worked on basically epigenomic modulation in cortical plasticity during adolescence, but also in neurodevelopmental disorders like Rett syndrome. And he's now working as a project scientist at the Glass Lab at UCSD, where he's exploring the epigenome of the human brain in order to have more understanding on the gene variation contribution to neurodegenerative diseases like AD. And he recently had a paper in Science where they basically dissected several promoter enhancer interactomes in different CNS cell types from human tissue. And I guess I'll leave you to Dr. Alexi Nott to give us more insight on that work. We're really happy to have you.
Great. Okay, thank you for the nice introduction. Okay, I'm just going to share my screen. Okay, so first of all, I'd like to say, great. So yeah, I'd first like to say thank you for the introduction, and also thank you to the NeuroMac initiative for inviting me to give this seminar today. As Omar mentioned, I'm working in the laboratory of Chris Glass at University of California, San Diego. So I'm on the West Coast. And the project I'm going to present today was a collaborative effort with a bioinformatician in the lab called Inga Holtman, and also with another assistant professor at UCSD called Nicole Kupal, who did a lot of the iPSC work that I will be presenting at the end of my talk. So in the Glass Lab, we are interested in looking at the role that, or information that non-coding regulatory regions can give us to try and explain the genetic risk for different brain disorders. So with a focus on Alzheimer's disease. Just an overview of what I'm going to go through today. So I'm going to basically go through the paper that was recently published, which was on brain enhancer promoter connectivity maps and how they help provide information on disease risk association. And as a brief summary, the major findings from the paper, we found that microglia enhancer regions or gene regulatory regions are enriched for DNA variants that are associated with an increased risk of getting Alzheimer's disease. And then we use chromatin looping to be able to tie these disease risk variants to the target genes. So as a way of an introduction to Alzheimer's disease, so dementia is the sixth leading cause of death in the US, and Alzheimer's disease is the most common form of dementia. And for a small subset of individuals who have for a small subset of individuals who have Alzheimer's disease, these individuals have an early onset form of the disease, normally occurring when they are 65 years or younger. And for these individuals, there's normally the mutations are known that are causative, so either mutations in APP, presenilin 1 or presenilin 2. Which led to the amyloid cascade hypothesis. However, these individuals constitute less than 5% of people who have Alzheimer's disease. The vast majority of people, if you look at the pie chart on the left, have a later onset version of the disease. So it occurs when they are either 75 or 85 years and older. And for these individuals, so these individuals constitute over 95% of people who have the disease. And the underlying genetics for these people is unclear. So this graph is just to illustrate that by 2050 is expected that the aging population in the US is going to double in size. So this is just to kind of highlight the health crisis that the US is facing in regards to aging-related disorders, including Alzheimer's disease. And this is not just true for the US; it's also true for other places in the world. So this creates a kind of a need to better understand these diseases, so we can come up with new treatments. So despite the fact that the underlying genetics are unclear, there's been a number of genome-wide association studies that have started to be done for these different brain disorders. So these studies basically sequence many people who have the particular disorder you're interested in. So in this case, Alzheimer's disease, and they sequence common variants in these individuals, and then they sequence common DNA variants in age-matched control people. And they look for DNA changes in the genome that are associated with an increased risk of getting these diseases. So this has been done for a number of different brain disorders, including in 2019. So in this year, there were 2 GWAS studies that came out for Alzheimer's disease. And with these GWAS studies, we now have a large number of DNA variants that are associated with an increased risk of getting the disorder. And these are located across 30 different loci in the genome. So one of the issues in interpreting GWAS studies, which I'll get to in a bit more detail later, is that these variants are often in non-coding regions, which has meant that in many cases, the genes that are assigned to these loci is based on the nearest gene to these non-coding DNA variants. So the gene allocations provided in this figure are quite often based on the nearest gene hypothesis. So okay, and then so going back to data that was produced from the GLASS lab in 2017. So the GLASS lab has an interest in looking at the gene regulation in microglia in the brain. So in a study done in 2017, which was led by David Gosselin, he looked at the transcriptome of human microglia isolated from resected tissue from the cortex. So these were microglia that were isolated from epilepsy patients. So the tissue that David was using was trying to avoid the epileptic foci, and basically the microglia that we were isolating were they are as close to control or healthy microglia that we can get with the currently available material. And David looked at the transcriptome of these microglia, and using the GWAS assigned risk genes for Alzheimer's disease, he compared the gene expression in microglia to that of cortical tissue. So that's bulk tissue that's not been, there's no cells being isolated from them. And he found that there was high levels of gene expression for these AD risk genes in microglia compared to whole cortex, implying at this point that perhaps microglia might be an important cell type for driving Alzheimer's disease risk. However, as I alluded to earlier, there are a number of challenges in interpreting genome-wide association studies, or GWAS studies. So one of these issues is, again, that the variants are non-coding, and also they tend to cluster in these clouds that cover loci that include more than one gene. So one of the issues, or one of the challenges, is determining which genes are affected by these DNA risk variants. Another issue is that, or challenge, is that GWAS studies are done using genomic DNA. So they provide no information on the cell types that these DNA risk variants are acting on. And for a complex organ like the brain, which is made up of multiple cell types, another challenge is determining which cell types are affected by these DNA risk variants. And the fact that these variants are often non-coding has led people to hypothesize that they might actually be regulating they might be altering the function of gene regulatory regions called enhancers, which is something that the GLASS lab is particularly interested in. So enhancers are short regions of DNA that are distal from the gene, so they're far away from, well, not far away, but they're distal from the gene, and that upon binding transcription factors, they form these DNA loops that bring them into close proximity to the gene promoter and create an environment that's permissive for gene expression. However, so the hypothesis is that the location of these disease risk variants within these enhancer regions may either affect the ability of transcription factors to bind to these enhancers, or in some way may affect the ability of these enhancers to loop to their target gene promoters and thereby alter gene expression. So this could be either an increase in gene expression or a decrease in gene expression, depending on how these risk variants are affecting the ability of transcription factors to bind or altering the types of transcription factors that are binding. Okay, so when I started this project, the enhancer landscape of different cell types in the brain had not been systematically looked at. So to begin to address this problem, I took a nuclei isolation approach where I isolated nuclei from cortical tissue, again from epileptic patients. So I used a similar cohort of patients or individuals as was used in the original 2017 paper by David Gosselin. And then I stained these nuclei for cell type specific nuclear markers and sorted them or isolated them by fluorescence activated nuclei sorting. And these cell type specific nuclei populations were then subjected to a series of genome-wide sequencing assays. So just to summarize the cell types that I was looking at, I was able to isolate nuclei from microglia by staining for the transcription factor PU1, as you can see on the far left. And then moving along, I was able to isolate neuronal nuclei by staining for the splicing factor NeuN, oligodendrocyte nuclei by staining for the transcription factor OLIG2, and astrocytes were isolated by using a combination of two marks. So I was sorting on NeuN negative and LHX2 positive nuclei. So these LHX2 is another transcription factor. And then taking these cell type specific nuclei populations, I then performed a series of sequencing assays, which included ATAC-seq. So ATAC-seq identifies open chromatin regions. And these are regions in the genome that we hypothesize are where transcription factors are likely to bind. We then also did a number of chip sequencing or chromatin immunoprecipitation sequencing assays for two histone marks, so for H3K27 acetyl and H3K4 trimethyl. So the H3K27 acetyl histone mark identifies regions on the genome that are either active promoters, which is this region highlighted in pink for the TREM2 locus, or active enhancer regions, which is highlighted here in yellow, distal from the TREM2 gene. Conversely, H3K4 trimethyl predominantly marks only active gene promoters. So you can see here in pink, you see H3K4 trimethyl signal. However, there's very little signal in the enhancer region highlighted in yellow. So by using a combination of these two marks, we're able to distinguish between active, what we think are active gene promoters versus active enhancer regions. Okay, so first of all, we wanted to check the cell type specificity of our data sets. So to do this, the examples given here are for cell type marker genes. So here I'm showing, which I'm sure you're all familiar with, is CX3CR1. So it's a typical myeloid marker. And you can see that the microglia tracts here shown in red shows signal for the CX3CL1 for the ataxic H3K2707 and the H3K4 trimethyl. And there's very little signal from the other cell types that we looked at. So very little signal from the neurons or oligodendrocytes or astrocytes. And we see a similar pattern here in green for the neuronal marker neurofilament light. Again, for an oligodendrocyte marker shown here in blue. And on the far right, a similar pattern for an astrocytic marker shown here in orange. So the examples I've given here are one particular cell type marker for each cell type. However, we wanted to do this more systematically in genome-wide, so computationally in genome-wide. So to do this, we looked for active gene promoters that were active in one cell type and not the other three. So if you look at the top left-hand corner of this heat map, you can see a number of active gene promoters that are active in microglia and not in neurons, oligodendrocytes, or astrocytes. This is followed by gene promoters that are active only in neurons. After that, gene promoters that are active only in oligodendrocytes, and then finally, those that are active in astrocytes. And then we wanted to compare this to RNA-seq data that was generated for each of these cell types by Ben Barris's lab in 2016. And you can see that they did RNA-seq on these cells. Sorry, they did RNA-seq analysis on these same cell type populations, and you can see that the gene expression analysis looks very similar to what we find for the active gene promoters. So indicating that the cell types that we're isolating are really the cell types that we think they are. We then took these active gene promoters, and for the genes associated with these promoters, we did, okay. And sorry, my apologies. So we took the genes associated with these active gene promoters, and we did a gene ontology analysis. And we found that for active gene promoters in the microglia data set, we saw gene ontology terms associated with myeloid cells. We saw neuronal gene ontology terms associated with active gene promoters in the neurons. And we saw glial gene ontology terms for active gene promoters that were found in the oligodendrocytes and the astrocytes. So going back to the slide I showed before, we used a combination of the H3K27 acetyl and the H3K4 trimethyl to annotate across the genome regions of active gene promoters and active enhancers for each of the 4 cell types. So this is summarized here for the promoters. So this is like a Venn diagram. And you can see that while there is a number of cell type specific active gene promoters for each of the cell types, the vast majority of these active promoters are actually shared in common between all 4 cell types. In contrast, when you look at the active gene enhancers, first of all, you will note that there's many more active gene enhancers compared to promoters. So this is a many-to-one relationship, which had actually been shown before. So there's more enhancers for every for every gene that's expressed. And another interesting observation was that the cell type specificity is a lot greater at the level of the enhancers than it is of the promoter. So there's a lot more cell type specificity in the enhancer landscape than there is in the promoter landscape. So this indicated to us that a lot of the cell type specificity in gene regulatory programs is driven rather within the enhancer repertoire rather than being driven by the promoter. Okay. We then wanted to look to see if there were DNA risk variants enriched within these enhancer regions. So we did this for a number of using GWAS data for a number of neurological disorders, psychiatric disorders, and behavioral traits. So I want to bring your attention to the red box to the left. So this is looking at 2 GWAS studies for Alzheimer's disease that came out this year. And you can see that DNA variants that are associated with an increased risk of getting Alzheimer's disease are enriched in the enhancers of microglia and not in the enhancers of the other 3 cell types. And interestingly, the microglia enhancers only showed enrichment for AD DNA risk variants and not for any of the other GWAS studies that we looked at. You can compare this to what is shown in the green box here for psychiatric disorders, that DNA risk variants for psychiatric disorders are largely enriched in neuronal enhancers. Examples being autism and schizophrenia, recapitulating, in the case of schizophrenia, what was found by the PsycENCO project. Okay, so we then wanted to be able to tie these gene regulatory regions associated with disease risk to their target genes. So to do this, we used an assay called proximity ligation assisted ChIP-seq, or PLAC-seq for short. So in brief, this assay involves first performing a proximity ligation. So this basically ties together 2 regions of the genome that are in close proximity to each other, followed by a chromatin immunoprecipitation for H3K4 trimethyl, which is the histone mark for active gene promoters. So by performing this assay, what we're doing is we're pulling down active gene promoters, and then by looking at chromatin looping, we're seeing what distal regions these active gene promoters are tied to. So by doing this, we're trying to tie enhancer regions to their target genes. Okay, and then an example of this is the SAL1 locus. So this is shown here. So the SAL1 gene itself, the promoter of the gene is highlighted in pink on the left. And the SAL1 locus also contains a microglia-specific super enhancer. So this is a super enhancer of the SAL1 locus that's only in microglia, not the other cell types. And if you look at the chromatin looping, and this super enhancer is highlighted in yellow. And if you look at the chromatin looping in the bottom track, you can see that there are microglia-specific chromatin loops that tie the super enhancer region to the SAL1 gene in pink. So we then wanted to again look at the cell type specificity of this data. Oh, sorry, before that, this is a summary of the kind of information that we got from these assays. So for oligodendrocytes, we were able to see over 60,000 PLAC-seq interactions, and we were able to see around 100,000 PLAC-seq interactions in the microglia and the neurons. So we did it for 3 of the 4 cell types. And by looking at whether the plaque interactions were, what gene annotations were associated with these plaque interactions, we found that the majority fell within either promoter-promoter interactions, promoter-enhancer interactions, or promoter-attack-seq interactions, indicating that the majority of the plaque-seq interactions that we were getting were likely to be functional. We then wanted to see, again, check the cell type specificity of our data sets. So here, similar to before, we're looking at plaque-seq interactions that are specific to either microglia, shown at the top, followed by plaque-seq interactions that are specific to neurons, and lastly, plaque-seq interactions that are specific to oligodendrocytes. And then we took the genes that are associated with these cell type specific plaque-seq interactions, and we did gene ontology on these genes. And we found that genes associated with microglia specific plaque-seq interactions had myeloid gene ontology terms. And as expected, we saw neuronal gene ontology terms for those genes associated with neuron-specific plaque-seq interactions, and lastly, glial terms for glial ontology terms for the oligodendrocyte-specific plaque-linked genes. We then wanted to be able to use the plaque-seq data to see if we could get some information regarding Alzheimer's disease risk variants. So we saw a couple of features when doing this analysis. So one of the examples is given here for the locus PTK2B and CLU. So PTK2B and CLU have already been assigned as GWAS risk genes for Alzheimer's disease. And this is an example of where our PlaqueSeq data validates the assignment of these genes by the GWAS study. So we see that PTK2B and CLU are likely to be AD risk genes. However, by using our plaque interactome, we are able to identify additional AD risk genes at this locus. And in this case, we see an additional 4 genes. And also, we're able to identify which cell types that they're likely to be affected by these DNA risk variants. So this is an example of where we can extend the number of AD risk genes for a particular locus. Here's another example for the SLC24A4 gene. In this case, the AD risk variants are highlighted here in turquoise on the far right, or in green. And you can see that the AD risk variants are located within the intron of the gene SLC24A4. So this gene has been described as the GWAS risk gene for this locus. However, you can see that these variants fall within what appears to be a microglia-specific enhancer region. So there's a TAC-seq signal and H3K27 acetyl signal in microglia and not the other 2 cell types. And if you look at the bottom track, you can see that this microglia-specific enhancer region that contains these AD risk variants is actually tied to more distal gene promoters, which includes the ataxin-3 gene. And importantly, it's not tied to the promoter of the SLC24A4 gene, which is highlighted in pink. So this is an example where we are able to reassign the risk genes for a particular locus. You can also see from the pink highlight, which is the promoter region of the SLC24A4 gene, that this gene seems to actually appear to show very little expression in any of the cell types that we looked at, compared to the more distal gene targets. Okay. We then did this type of analysis more systematically across all the AD risk loci. And we identified 73 genes that were plaque linked to Alzheimer's disease risk variants across all the different cell types. And of these 73 genes, we found 50 genes were in microglia. And of these, 25 were specifically in microglia and not the other cell types. So this led us to believe that microglia might be an important cell type for driving some of the genetic risk of getting Alzheimer's disease. This type of analysis also allowed us to come up with a gene network for Alzheimer's disease that's predominantly genes that are expressed in microglia. Next, we took these plaque-linked AD risk genes for each of the cell types, and we did a gene ontology analysis. And we saw gene ontology terms associated with mainly immune functions, as you would expect, particularly in the microglia AD risk genes, and also for amyloid processing. So the amyloid processing gene ontology terms were shared for AD risk genes in neurons, oligodendrocytes, and microglia. So lastly, we wanted to be able to validate our approach. So to do this, we chose the BIN1 locus. So the BIN1 locus is interesting for a different reason. So BIN1 is interesting because it's expressed in all 4 cell types in the brain. So this has been shown if you look at Ben Barris's RNA-seq data for cell types in the brain. And also if you look at our promoter analysis, you can see that BIN1 has active promoter marks for microglia, neurons, oligodendrocytes, and astrocytes. So the promoter region is in these tracks is highlighted here in pink. And you can see that there's H3K4 trimethyl marks and H3K27 acetyl marks for each of the cell types in the promoter region of BIN1. However, the enhancer landscape for BIN1 is distinct between each of the cell types, indicating that how BIN1 gene expression is regulated is unique for each of the cell types. So an example highlighted here in yellow is a microglia-specific enhancer region for the BIN1 locus. So this enhancer region has a tag-seq signal and H3K27 acetyl signal in microglia and not the other 3 cell types. And this enhancer region is interesting because it also happens to contain the lead AD risk variant for this locus. So what we believe is likely to be the causal variant for the BIN1 locus is within this microglia enhancer specific enhancer region. And if you look at the bottom tracks, you can see this microglia enhancer region is also plaque linked to the BIN1 gene. So we believe that the target gene for this enhancer is BIN1. So to begin to validate this, so this is a magnification of the enhancer region. And again, you can see, first of all, that there's H3K27 acetyl and a tag-seq signal specifically in the microglia and not in the neurons, oligodendrocytes, and astrocytes. And to begin to validate this enhancer region, Nicole Kufal used CRISPR to delete a small region of the enhancer that encompasses the tag-seq peak. So the tag-seq peak is where we think the transcription factors are likely to bind. And in addition, this deletion region also includes the lead AD risk variant that I mentioned before. So what we believe is to be the likely causal variant for the BIN1 locus. And she deleted this enhancer region in iPSCs, so induced pluripotent stem cells. So we had the microglia-specific enhancer deleted iPSCs, as well as control iPSCs. And these iPSC cells were then derived into 3 different cell types, into astrocytes, microglia, and neurons. So images of each of the cell types are shown here on the far right. And then we wanted to look whether BIN1 gene expression was altered in each of these cell types. So we did this by RNA-seq analysis. So first, I want to point out that BIN1 gene expression in the control cells. So if you look either on the tracks on the left or the gene expression data is summarized also on the right in the bar graph, you can see in the gray bars, which is the BIN1 gene expression in the control cells, that BIN1, first of all, is found to be expressed in the iPSCs, the microglia, the neurons, and the astrocytes. However, when you use cells that have the microglia-specific enhancer region deleted, you can see that the BIN1 gene expression remains unaltered in the iPSCs and is unaltered in the neurons and unaltered in the astrocytes. However, BIN1 gene expression is highly reduced specifically in the microglia. So kind of validating the fact that this really is a microglia-specific enhancer region. So this data is interesting also because at this point, BIN1 was known to be an AD risk gene and had predominantly been studied in the context of neurons up until this point. So we believe our data, this is an example of how our data can provide cell type specific information. So going forward, we now believe that to study BIN1 in the context of Alzheimer's disease and aging, that microglia are an important cell type to consider. And up until now, the role of BIN1 in microglia is largely understudied. So we also confirmed these findings at the protein level. So if you look at the Western blots on the left-hand side, the Western blots at the top show BIN1 protein expression or protein levels in microglia that were derived from the control iPSCs or the iPSCs that had the microglia-specific enhancer deleted. And you can see that with deletion of the microglia-specific enhancer, that BIN1 protein levels are a lot lower compared to controls. However, if you do the same experiment, but this time the iPSCs are derived into neurons, you can see that there's very little difference in the protein levels of BIN1 between the control cells and the microglia-specific enhancer deletion. So the protein analysis is summarized on the right here in the bar graph. And you can see, again, that there's a strong reduction in the protein level of BIM1, specifically in microglia and not in neurons or astrocytes. So I just wanted to highlight here at the end that, again, these data sets were derived from young individuals who are epilepsy patients. So these data sets were the closest we were going to get to healthy controls. And for a number of AD risk loci and also for other brain disorders, there are loci that don't show gene regulatory regions that are active in any of the cell types that we looked at. So this could be for a number of reasons. One of the reasons might be that, first of all, we're not looking at the correct cell type. So there are additional cell types that we didn't look at that are in the brain, including endothelial cells. Another reason that we're interested in is that some of these might be context dependent. So they might be dependent, for example, on cell state as well as cell type. And some of these genomic loci might only become activated upon aging or in the disease state. So one area of interest in the lab is to now look at post-mortem samples to see how the epigenome and the interactome is changing in each of these cell types in disease situations. So in summary, what I told you today is, what I showed you early on, is that a lot of cell type diversity and specificity of gene regulation it occurs at the level of the enhancers rather than the promoters. We found that disease risk variants for Alzheimer's disease were enriched in microglia enhancer regions, whereas disease risk variants for psychiatric disorders were enriched in enhancers for neurons. Using our interactome maps, we were able to tie these disease risk variants to their target genes. And lastly, for the BIN1 locus, we were able to validate that a microglia-specific enhancer affects gene expression of BIN1 specifically in microglia and not in neurons or astrocytes, highlighting the cell type specificity of our data. And then this leads me to lastly thank everyone who was involved in the project. This involved work from a number of different individuals. Again, I want to highlight Chris Glass, whose lab I've been working in for the past 3 years. Inga Holtman, who was the lead bioinformatician on this project, and Nicole Kufal, who was instrumental in the iPSC work. I want to also highlight my funding sources. So I received a fellowship from the Alzheimer's Association. In the last year, I was awarded a Career Development Award, a KL2 Career Development Award from NIH at UCSD. And lastly, I also received some money for a pilot project from the Charlie Marcos Alzheimer's Disease Research Center at UCSD. And I am happy to take any questions.
Thank you so much, Alexi, for the nice talk. And so there are already some questions on the chat. Maybe some of them are technical. We can maybe go shortly through them. So the first question was from Jana, and she was asking about the sorting and was wondering if the stainings were intranuclear stainings.
Oh, so the majority of the stains were transcription factors, so they're intranuclear. So one of the first steps of the protocol is to fix the material, and then we use detergents to permeabilize them, and yeah, so it's an intracellular stain.
Okay, and then from the Max Planck, they asked if you also checked for other enhancer marks like K4 monomethylation. So
no, we have considered doing that, but we haven't done that yet. So we were actually thinking of looking at HBK4 dimethyl so that we could also identify primed enhancers. But we haven't, because of limitations in materials, we haven't got to that point yet.
I see.
But that's an interesting question. because at the moment we're basically just identifying the active gene, the active. Sorry, I'm mixing you guys up. So we wanted to look at the H3K4 dimethyl to look at poised enhancers. And with the data sets that we have at the moment, we're only really able to define active enhancers. So it would be interesting um, to do some additional marks, and that might also help explain some of these AD risk loci that are not that don't seem to have any active gene regulatory regions. Maybe they have some poised elements that we're missing in them. So yeah, I think that's an interesting question.
Yeah. And then regarding again the fixation and the internuclear staining, so Jen was just wondering how could you sequence fixed nuclei? So just regarding the technique.
Well, it's pretty standard for chip sequencing. So with chip sequencing, you always fix you always fix the material before you start, because you want to bind the protein targets that you're looking at to the DNA, and then you do shearing to cut up the DNA into small chunks, and then you do an immunoprecipitation, so you use antibody to pull out the regions that you're interested in. So the fixation helps to keep everything together. And then when you're making the libraries, you reverse cross-link everything. And then you chew up the protein using proteinase K, and then you purify the DNA, and you make your libraries with the purified DNA at the end. So you fix at the beginning.
That also worked for the ATAC sequencing, or do you think this also
sequencing it did what? So that's what I did. How did I do that? So I did I isolated the nuclei which were fixed. I did the tagmentation first, and then I reverse cross-linked before doing the PCR. But this is actually published in the ATAC-C SEE paper from Howard Chang's lab. Yeah. They do actually have a protocol written for that, but I have to warn you that you need good quality material to get that to work. So I'm not sure how well the ATAC sequencing will, using fixed material at least, will work on the postmortem samples. So part of the motivation of this project was actually to get high quality atlases to be able to compare to the postmortem tissue.
Yeah. And then basically the question was, what's the advantage of the plaque-seq compared to high-C?
Okay. So the advantage is that you can basically, so first of all, the high-C, so I should maybe explain to people the high-C, you're not doing any enrichment. So with the plaque-seq, as well as looking at the chromatin loops, you're also pulling out the active gene promoters. So you're first looking at active gene promoters and then looking to see where they're looping to. Whereas the high-C, you're just looking in more of an unbiased way, to be honest, where there are interactions across the whole genome, regardless of any other histone marks or any other kind of gene annotation. So it's true. One of the limitations, I guess, of the plaque-seq, or something you need to be aware of, is that there is a bias in the data, right? The data is driven in part by the presence of the H3K4 trimethyl promoter. The advantage of it in our situation is that the number of nuclei we can obtain is limited, right? We're not looking at cell lines and we don't have millions of cells. So the starting material that you need, because you're doing this enrichment step, the amount of starting material you need is lower. And then how deep you need to sequence is also lower. So it's also good if you want to include a number of different replicates. And then even with this lower starting material, from what I understand, you end up getting a lot more actual interactions to look at. Mm-hmm. So with the high-C, you have to sequence very, very deeply to be able to see these different interactions.
Thank you for the elaboration.
Does that answer the question?
I guess so. I guess they will write.
I mean, there's advantages and disadvantages to both methods. So I feel, so I guess my main point is, because of the limited material source and the fact that the plaque-seq uses an enrichment step, that was the main advantage for us to be able to use the plaque-seq.
Thank you. And I personally have a question. I guess you touched on it a bit at the end, but I was wondering how did it actually look like for genes that are usually passed around in the field, like ApoE or CD33 or CD11C, for example. I know you mentioned it shortly at the end, but I would like if you maybe can highlight a bit more.
So the ApoE locus, I'm not sure I could really explain it over the webcam because it's like a very complicated locus that's is highly interactive in terms of connections.
Yeah.
So the ApoE locus is actually quite difficult to analyze. And actually, if you look at the summary table, there's many GWAS like that. A lot of the risk genes that pop up within the ApoE locus. So and that's just because of how many interactions there are at that locus. CD45 and so for IGAM, I can't remember which is which, but we looked at IGAM and IGAC. So those are the gene names for those products.
Yeah.
And we found that I don't think we showed this in the paper actually, but there are plaque-seq interactions that tie those genes to AD risk variants. And this is also was touched on in the Janssen, I think it's the Janssen GWAS study. So if you look at that study, for one of the two genes, I can't remember if it was IGAM or IGAX, they also are able to tie, I think by using QTL analysis, that one of the two genes might be affected by Alzheimer's disease risk variants. I don't know, does that answer your question?
Yes, it does a bit. So we'll take some questions from some of the audience here. I'll just let them ask them themselves.
Hello, thank you for the very nice talk. I was
Hello.
wondering, now that you found out which genes are actually regulated or have issues, are you actually following up on the different functionalities of those genes in the specific cell types?
So because we have the system set up, so Nicole is actually in the process of looking at the functional consequence of the BIN1 enhancer region. So something that was not really highlighted in the paper was that, which is also why I kind of mentioned the disease context at the end, is that when we did the RNA-seq expression on the BIN1, so the microglia specific enhancer, where we see like BIN1 is almost completely disappeared in the microglia versus the controls. The only differential gene that we saw expressed was BIN1. So when you have iPSC-derived microglia sat in a dish, you don't really see much difference at the gene expression level between the two cell types, other than the target gene, right? So one reasoning is that this might be because it's the fact that the cells are sat in a dish and they're not in the correct environment. So going forward, we're going to start to look at how these cells behave in more of a disease context. So using through a collaboration with a group at UCI, Matthew Blood and Jones, these microglia, these BIN1 enhancer deletion microglia will actually be transplanted into mouse brains, and including control and Alzheimer's disease models to see how they behave in more of an in vivo setting. So that's one of the directions that the lab is going in regards to looking at the function of these risk genes.
Sounds really interesting.
But obviously, this could be expanded to other loci and other genes. But each of these is a project in itself, right? Each of them is going to take up a lot of work.
So we'll take one more question.
Hey, first of all, thanks for your nice talk. I had a question because I'm not familiar with these techniques. So can you maybe explain how stable these interactomes are between the single individuals you analyze? So basically, would you find the same interactions between enhancers and genes in all individuals, or are the changes? Is it sensitive enough, the method? Which then also relates to basically maybe if you look at a diseased individual, can you maybe see what changes there are compared to healthy controls?
So that's an interesting question. I'm not sure. So the way the analysis was done, we were looking at interactions that were fairly reliable between the individuals. So one of the limitations of this study is that, particularly for the plaque interactome data, the number of individuals we used was fairly low. So we used 3 individuals per cell type. So with such a limited group, it's kind of difficult to answer that question. But as we add more individuals and more samples, I think we would hope to be able to see differences in the interactome. To be honest, because of the sensitivity of the assay, I feel like we're more likely to see differences at the enhanced activation level. So at the level of histone acetylation, rather than probably the interactome, just because of the sensitivity of the assay. Okay.
Thank you again, Alexi. Someone else maybe have more questions? So I think if there are not any more questions, I would like to thank you again, Alexi, for the time, and thank also the people who attended the webinar here and the Max Planck. Oh, maybe someone is. Oh, okay, yeah. So also thank you from the Max Planck people. It was really nice having you, and thank you for dedicating this time for us and highlighting this great research. I think it's really revolutionary, and I think it paves the road for a lot more research in regard of the disease context and connecting the epigenomic studies more in depth and how they affect the function in a cell-specific manner.
Thank you for inviting me. It was a great experience. It was my first webinar, so
that's cool.
Nice. Thank you. Yeah. Thank you very much.